Hi dear students, welcome back to one more session of microbial interactions, and we had come across the basics of microbial interactions in the previous session, and now we are going to classify the microbial interactions into positive as well as negative interactions, and we would be sequentially learning what are the characteristic features of different types of interactions which are possible in the case of microbes. The interactions which give a positive benefit is referred to as a positive interactions, and they mainly include mutualism, syntropism, commensalism as well as protocooperation. And there are also certain interactions which have been carried out by the microbes and which will give a negative impact. They are referred to as the negative interactions, and we have examples of predation, parasitism, competition as well as commensalism. Now, today we are going to talk about the mutualism or the symbiotic relationships which have been carried out by microbes. Now, here this is a relationship in which most of them get a reciprocal benefit as a result of the reaction. That is, both the partners will get a positive interaction or a reciprocal benefit by having an interaction between each other. And here it is an obligatory relationship in which the mutualist and the host are dependent upon each other. Now, what do you mean by obligatory? Obligatory is something which has been found to be a must interaction. That is, without that particular interaction, they will not be able to survive, and when they are being found to be separated. So, the association of the both the members is a must for either of the partners, because when they are being separated they cannot live individually. So that is a case of mutualism or a symbiosis. Now the most classical example of symbiosis which we mentioned is in the case of lichen. In the case of lichen, it is a symbiotic relationship which is happening between a algae and a fungi. Now this is an algal component which has been schematically represented over here, and you also have a fungal component over here. And this is a picture of a lichen which is being shown over here. And as we all know, lichen is not a single organism, and it is a stable symbiotic association between a fungi and an algae or a cyanobacteria. And like all fungi, the lichen fungi will require carbon as a carbon source, as a food source. And this has mainly been provided by the algae or the cyanobacteria, which is considered to be photosynthetic. And the lichen symbionts is thought to be a mutualism, since both the fungi and the photosynthetic partners, called the photobionts, both of them will benefit from it. Usually, the lichen is made up of two or more closely interacting organisms, a fungi, and one or more partners called the photobionts. What is the photobionts? The photobiont is a one, either it can be a cyanobacteria or it can be a algae. Okay, so this cyanobacteria or the algae will mainly photosynthesize, and it will provide the what? Food towards the fungi. And the fungi is heterotrophic, and it will require an external source of food. And the fungi will build the structure of the lichen thallus, within which they will provide conditions for a long-term stable association with the either algae or the cyanobacteria. So the role of the fungi over here is to provide a thallus-like environment where the algae can multiply. So this is a case of a symbiotic relationship which has been happening between a algae and the fungal element. Now, which fungi can be used to form lichens? Usually, it is mostly been found to be that cup fungi or ascomycetes are the ones which have been involved in lichens, and many unrelated and very different fungi form lichens, including the mushroom forming fungi and especially the cup fungi. And fully half of all ascomycetes and 1 in 5 of all known fungi can form lichens. And lichenization is an ecological strategy or a common nutritional mode among the unrelated fungi. Now, what are the photobionts or what are the photosynthetic elements in these lichens? It can either be green algae or it can be cyanobacteria. In most of the cases, like in 90% of all the lichens, you can see that it is a green algae which is the photosynthetic partner over here. And in the remaining, in the remaining lichens, you can find cyanobacteria can also be found. So this is a case of symbiotic or a mutualistic relationship in which both the partners are being equally benefited and they cannot survive when they are being separated from each other. Of course, we also have various other examples of symbiotic relationship in with nitrogen fixing bacteria with plants and all that are there, and we will be coming across these examples and in the respective sessions of nitrogen fixation. Now, I would like to talk about insect-microbe mutualisms. One type of mutualism which we come across is in the case of protozoans as well as the termites. In the case of termites, now a worker, this is an example of a worker termite of Reticulotermus. It mainly eats woods. And inside this, you can find that there is a protozoan called the Trichonympha. And it has been found to be this is a picture of Trichonympha which has been found inside this termite, in the termite's gut. And you can see that the presence of this protozoan in the termite will enable it to break down the cellulose and it will allow the termite to use the wood as a food source. So by the relationship or by the attachment of the protozoan with the termite, what happens? The termite is enabled to utilize the particular wood in which it is being feeded or on which it grows on. So now in this symbiotic relationship, you can see that, of course, we told that the protozoan is producing some cellulases and the cellulases is being used to degrade the wood. We can see that when the cellulose is being broken down by the trichonympha using the particular cellulases, it gets converted into acetate as well as other products. And these products can be further being utilized by the termites. And the termites will thus get a good source of energy for its multiplication or for its survival. Because the termite as such, it cannot use cellulose because it does not have the enzyme cellulases. Now, when this is being utilized by the termite, what happens? It will also produce a highly anoxic environment and the protozoan here, which is a protozoan, the trichonympha, as we are talking about here, that is being found to be highly anoxic in nature. So the when energy products which are being produced by this trichonympha is being utilized by the termite will produce a highly anoxic environment so that the trichonympha will get a highly hospitable environment for its multiplication, as well as it also gets a hose. So the wood is being eaten up by, has been taken by the termite, but the enzymes for the degradation of the cellulose is being provided by the trichonympha, and trichonympha will break down the cellulose into acetate as well as various other end products which can be utilized by the termite, and the termite, as it utilizes the acetate products, it will produce a highly anoxic environment so that trichonympha also can multiply within it. So this is a case of a symbiotic relationship which has been found between a protozoan as well as a termite. Now another example which we have here is a coral and Zooxanthellae. You can see that various marine invertebrates like sponges, jellyfishes, sea anemones, corals as well as ciliated protists, they will have various endosymbiotic dinoflagellates called the zooxanthellae within their tissue. Now you can look into this picture over here. This is a hydra's tentacle, and inside that you can find some green color zooxanthellae. And the green color of the rose, this is another coral. Here also you can see that a green color dinoflagellate is being found as an endosymbiont within the particular tissue. If you take a close look onto the coral and zooxanthellae association, you can find that these reef building corals, most of the if you go to look at them, you can see that most of the energy requirements is mainly being met by the zooxanthellae, because zooxanthellae is capable of photosynthetically fixing carbon, and much amount of carbon which is being needed by the coral reef is being provided by the zooxanthellae. Now what does the zooxanthellae get benefited by this? You can see that these algae are being protected from the UV rays using the pigments of the corals. So in turn, you can see that when zooxanthellae gives fixed carbon to the corals with its pigments will protect the algae from the harmful effect of the UV rays. And you can see that in exchange, zooxanthellae will also receive various nitrogenous compounds, phosphates as well as carbon dioxide also, in addition to the UV light from their hosts. And of course, you can see that the zooxanthellae also benefit the coral as the rate of calcification is being found to be greater in the light than in the dark. But you can see that right now, there are also cases of coral bleaching which is happening, and this is mainly defined as a loss of the photosynthetic ability of the zooxanthellae, which has been happening in the coral reefs. Now, it can be found that by the process of global warming and all that, these zooxanthellae are losing their photosynthetic pigments, and even a small temperature change of about 2 degrees centigrade will cause a removal of these zooxanthellae from the corals. And as a result, what happens when the zooxanthellae is being lost from over there, the carbon source to the corals are also being depleted, and the corals are getting bleached. And we can find that when there is a coral as well as an algal mutualistic relationship, there occurs the process of capturing, conserving and cycling nutrients as well as energy is being possible. That is, all these processes of conserving and capturing as well as cycling of nutrients and energy is possible when the corals and the zooxanthellae are being found to be associated with each other, and the coral reefs are being considered to be as the most productive and successful of the known ecosystems when they are being associated with zooxanthellae.